Hello everyone and welcome to Lecture 7. We're going to talk about a few common intracellular signaling pathways. In other words, what happens downstream of G protein coupled receptors? So we're going to start with G proteins. We'll talk about some secondary messengers, and then we'll touch on phosphorylation, which is a very important post-translational modification. We'll end with a little bit of gene expression. Alright, we've talked quite a bit about ionotropic receptors. Let's give metabotropic receptors the attention that they deserve. The metabotropic receptors are going to stimulate intracellular signaling cascades using one of about 3 different G proteins. So a G protein is a protein that binds to a guanine nucleotide. So a G protein coupled receptor is thus a receptor that's attached to a G protein. So G proteins have 3 different subunits. There's the alpha and then the beta and gamma. Beta and gamma tend to work together as a unit, so we kind of think of them as one and the same. The alpha, on the other hand, operates independently. And whether or not the G protein is active is determined by which guanine nucleotide is it bound to. When bound to GDP, or guanine diphosphate, the G protein is inactive. Once it binds to GTP, or guanine triphosphate, that causes the alpha subunit to dissociate and become active. The beta-gamma complex also becomes active, but it does not move around within the cell. The beta-gamma complex instead is buried in the membrane. The cell membrane is shown on the top of this illustration in orange. The alpha subunit, once it binds to GTP, is going to then leave the membrane and activate some sort of signaling pathway within the cell. Now, G proteins, because G protein coupled receptors have to use these intracellular proteins to create some sort of cellular response, it's good to keep in mind that metabotropic response is always slower than ionotropic responses. That's why we spend so much time on ionotropic channels, because they mediate that fast synaptic signaling that's going to really dominate a lot of the everyday signaling. The metabotropic component, though, is very, very important because it leads to long-lived changes in cellular function. We cannot forget homeostasis, of course. There always needs to be some regulation built in, and G proteins have self-limiting properties because the alpha subunit is a GTPase. In other words, it's a protein that hydrolyzes GTP to form GDP. It turns itself off as a result. So after the alpha subunit gets activated, once it's bound with GTP, it's only a matter of time until the alpha subunit acts on GTP and breaks it down. Once it reforms GDP, that causes the alpha subunit to move back to the membrane and associate with the beta-gamma complex. Now, of course, the balance between GTP and GDP is going to be regulated by other proteins. Some of them are going to speed up the rate at which GTP is hydrolyzed. These would be the GAPs, or the GTPase activating proteins. So they're going to help turn off G proteins by stimulating the conversion of GTP to GDP. So a GAP will turn off the alpha subunit. GEFs, on the other hand, the guanine nucleotide exchange factors, facilitate the exchange of GDP for GTP. So a GEF will help the alpha subunit swap out the used low energy GDP in place of a higher energy GDP. So GEFs will turn on G proteins, whereas GAPs turn them off. Now, what happens under the hood away from the membrane totally depends on which alpha subunit is being stimulated. That's what determines what G protein we're dealing with. It's the alpha subunit. The beta-gamma complex, they're the same. The alpha subunit is what differs. And there's really 3 types. There's GS. The S stands for stimulates. And what's stimulated is adenylyl cyclase. We'll talk about that in just a moment. GI or GO, they're the same thing. They inhibit. So GS stimulates, whereas GI inhibits adenylyl cyclase. So same enzyme, different effects. And then there's GQ. And in this case, Q, of course, stands for stimulates. A different target, though. Now we're targeting a different enzyme, phospholipase C. Okay, so we have two different enzymes that we now have to think about. We'll start with adenylyl cyclase. So this is turned on by GS and turned off by GI. Okay, adenylyl cyclase is an enzyme that's going to help generate cyclic AMP. Okay, so it's going to take ATP, that high energy molecule that the cell uses to do everything, and it's going to convert it into cyclic AMP, which we can see in this illustration here. Cyclic AMP is going to be an important secondary messenger. It's going to act on a few different targets. More on that later. And so remember, if we have a means of turning something on, we need to have a means of turning it off. And so there are proteins called phosphodiesterases, which break down cyclic AMP. They break that cyclic bond there in the phosphate group and turn it into regular old AMP. So what makes this cyclic is the fact that phosphate there is connected completely to that ribose sugar there. So the adenylyl cyclase is going to be responsible for creating cyclic AMP. Phosphodiesterases break down cyclic AMP. And then whether or not adenylyl cyclase is active or inhibited depends on the G protein. Okay, so if we activate adenylyl cyclase, cyclic AMP levels go up. If we inhibit it, they drop. And the opposite's true for phosphodiesterases. So we mentioned adenylyl cyclase. Now let's talk about phospholipase C. Okay, phospholipases are enzymes. You can tell that from the A's. They're going to act on phospholipids, and you can tell that from the phospholip part of the name. So phospholipase tells you exactly what it is. It's an enzyme that acts on phospholipids. So it's going to cut phospholipids in different regions depending on which phospholipase it is. Is it phospholipase A? Is it phospholipase C or D? In this case, we're going to be talking about phospholipase C. This is going to cut phospholipids in the membrane. And in this case, phosphatidylinositol bisphosphate. It's going to cleave it at the location shown here to liberate diacylglycerol and IP3. IP3 is inositol triphosphate. So that little 6-ring carbon shown there with 3 phosphates on it, on the 1st, 4th, and 5th carbons, that's inositol triphosphate. Inositol triphosphate is an important secondary messenger. It's going to stimulate calcium release. Okay, and the way that it does that is by acting on IP3 receptors. So the rough ER is, of course, very important for protein synthesis. Oftentimes, this is where the discussion of the ER ends. There's also the smooth endoplasmic reticulum within our cells that acts as an intracellular calcium store. So the calcium held inside the smooth endoplasmic reticulum only gets out if there's a particular message sent, and one of those messages would be IP3. So if we activate a GQ coupled G protein coupled receptor, then that GQ alpha subunit will activate phospholipase C. When phospholipase C is activated, it's going to liberate IP3 from the membrane, so it's going to cut phosphatidylinositol bisphosphate and create IP3. IP3 will float on down to the smooth endoplasmic reticulum and bind to IP3 receptors, causing the release of calcium. You get a very large release of calcium because there are also ryanodine receptors that are calcium sensitive calcium channels. So when you get a little bit of calcium release, that then causes what we call a calcium-induced calcium release. So you get a maximal release of all the intracellular calcium. It's kind of like the action potential, right? Once you get enough depolarization to activate a few voltage-gated sodium channels, then you activate them all. Okay, so cyclic AMP and that calcium that we just discussed, those are important secondary messengers, and they're going to activate a variety of different kinases. Now, a kinase is just a protein that's going to phosphorylate its targets, and more on this in the next main point. There are a few different kinases that come up time and time again. Protein kinase A is a very common intracellular kinase. The activity of protein kinase A is determined by the level of cyclic AMP. So protein kinase A has really two subunits, two types anyway. There's two catalytic subunits shown as C in this illustration. And then there's one regulatory subunit. It's a dimer, so it's actually two subunits fused together. And the regulatory subunit normally binds to the catalytic subunit and keeps it inactive. This interaction is disrupted when cyclic AMP levels increase. So in this case, in this cartoon on the right, they're showing you cyclic AMP. That's those little red triangles. There are binding sites on the regulatory domain for cyclic AMP. And when cyclic AMP binds, that causes the regulatory domain to change its shape and disallows the interaction with the catalytic subunits. So when cyclic AMP levels increase, the regulatory subunit releases the catalytic subunits and they move about the cell and then phosphorylate their targets. When cyclic AMP levels drop, the regulatory subunit no longer binds to them and instead binds to the catalytic subunits and turns them back off. Calcium is going to activate a different kinase. A very important kinase in neurons is CAM kinase 2, which is calcium calmodulin dependent protein kinase 2. It's a mouthful, so we just say CAM kinase 2. CAM kinase 2, just like protein kinase A, has a regulatory domain that's going to be inhibitory, and then it has a catalytic domain. And unlike protein kinase A, CAM kinase 2 is going to function as a multimer. So it's going to be 12 subunits together to function as one kinase. Now, this CAM kinase 2 is calcium calmodulin dependent. Calmodulin is another protein. It's a calcium binding protein. And so really calcium is going to activate CAM kinase 2 via calmodulin. So when calmodulin binds to calcium, that allows it then to bind to and displace that regulatory subunit in CAM kinase 2. And that's what will turn CAM kinase 2 on. So let's walk through this illustration. On the left-hand portion, we can see a single subunit of CAM kinase 2. So there's the regulatory domain. That's the kind of orange, floppy part there. And then there's the catalytic domain, which is shown colored. It's blue when it's off and it's green when it's on. So we have to remove that regulatory domain because it's sitting in the way of the CAM kinase 2 binding site for its targets. So when calcium calmodulin associates with the regulatory domain, that removes it from the catalytic domain and thus turns CAM kinase 2. The last protein kinase that we're going to discuss is protein kinase C. This is going to be activated by calcium as well as diacylglycerol. So that little bit of phosphatidylinositol bisphosphate that remains after IP3 is liberated. So protein kinase C is going to exist as a monomer, so just a single protein. There's the regulatory domain that is, of course, going to interact with the catalytic domain and keep it turned off, and that's going to keep protein kinase C inactive. Protein kinase C is going to be activated by calcium and diacylglycerol, as well as phosphatidylserine as well. But at the very least, you need calcium and diacylglycerol, which is what you get when a GQ G protein is activated. So phospholipase C cuts phosphatidylinositol bisphosphate, and creates IP3 and diacylglycerol. Diacylglycerol will bind to the C1 domain to increase the affinity of the next C2 domain for binding to calcium. So the C1 and C2, these are different parts of the regulatory domain. So after we've cut out IP3, diacylglycerol binds to C1 and increases the affinity of C2 for calcium binding. That IP3, of course, floats on down, stimulates calcium release, and then calcium can bind to the C2 domain and then prevent that interaction between the regulatory and the catalytic domains. Once that happens, protein kinase C is thus active because the regulatory domain is no longer keeping the catalytic domain inactive. This illustration is just showing you a few different mutant forms of protein kinase C that exist out there. Some of them actually lack the regulatory domains. For example, if you look at the bottom, this is what we call constitutively active protein kinase because it's always on. It lacks that regulatory domain, so it's only the catalytic domain. So that's protein kinase M, but it's actually just constitutively active protein kinase C there. Okay, so we've turned on these kinases, and what they do is phosphorylate targets. So they add phosphate groups onto target proteins. And so phosphorylation and dephosphorylation, these are very important modifications that occur to proteins after they're made. That's what a post-translational modification is. A change that occurs after translation, so after the protein is synthesized. Okay, you see these all over the place. You're going to find a lot of phosphorylation and dephosphorylation. What you should keep in mind is that phosphate groups are charged. When you add a charged group onto a protein, it's going to change how the protein folds up, and by changing the structure, you change the function of the protein. Kinases are just enzymes that are going to add phosphate groups. They'll add it to either a serine or threonine or tyrosine residue. A phosphatase is an enzyme that removes a phosphate group. That's it. So phosphorylation is carried out by kinases, dephosphorylation by phosphatases. So in this cartoon, we're looking at some protein that's getting phosphorylated and dephosphorylated by a kinase or a phosphatase depending on which receptor is activated. Okay, so some kinases are going to be bound to the membrane. Those would be what we call receptor tyrosine kinases. And almost without exception, these are going to phosphorylate tyrosine residues. So the receptor kinases almost always phosphorylate tyrosine residues. Once you move away from the membrane, though, and you're soluble, those kinases tend to be serine and threonine kinases. So they phosphorylate different amino acids when they're soluble. The activity of kinases and phosphatases, these have to work together because one is going to stimulate where the other is going to inhibit, and generally there's going to be some coordination built into these signaling pathways that allow them to work together. We've mentioned a few kinases. Protein phosphatase 1 is a very important phosphatase. The phosphatase inhibitors are generally activated by kinases. So, for example, here we have protein phosphatase 1 being inhibited by this regulatory protein DARP. So DARP32 is turned on by phosphorylation, so kinases activate it, thus inhibiting a phosphatase. The reason for this is that if you're activating receptors that are going to cause phosphorylation, you can amplify that signal by inhibiting the phosphatases that are there to inhibit phosphorylation. So whenever cellular signaling is shifting towards phosphorylation, this can be amplified even further by turning off phosphatases and vice versa. So there tends to be some way that phosphatases and kinases regulate one another. Okay, but the details totally depend on the specific signaling pathway. So at this point, all we can do is appreciate the fact that kinases phosphorylate, phosphatases dephosphorylate, and usually they can control one another. In this case, protein phosphatase 1 can be inhibited by kinases through that DARP32 protein. Now, whether or not the neuron is biasing itself toward the activation of phosphatases or kinases in some ways depends on the level of activity. And this comes back to calcium levels. Calcium is a very important secondary messenger. And so the concentration of calcium is going to affect whether we're biasing toward phosphorylation or dephosphorylation. So if we have a low level of activity, there's going to be generally lower levels of calcium, and so there'll be low levels of calcium calmodulin. Okay, calmodulin is that calcium binding protein. The low concentrations of calcium calmodulin will only bind to the highest affinity binding partner. In this case, it's going to be the phosphatase calcineurin. Calcineurin gets activated just like the other kinases we've talked about. It's the exact same cellular logic. Calcineurin is normally held in an inactive state by some regulatory subunit. When calcium calmodulin binds to the regulatory subunit, that inhibition is removed, and now we have active calcineurin. Calcineurin can then dephosphorylate kinases such as CAM kinase 2, for example. On the other hand, if you have a high level of neuronal activity and very high levels of calcium, then the lower affinity targets can be bound, like CAM kinase 2. So high levels of calcium calmodulin are going to activate CAM kinase 2. CAM kinase 2 can phosphorylate and inactivate calcineurin. So it doesn't matter what you want to do, you can bind all the calcium calmodulin you want. Once CAM kinase 2 phosphorylates calcineurin, all bets are off, and now we're biased toward phosphorylation. So very active neurons are going to have high levels of calcium calmodulin. They're going to be biased toward phosphorylation. Once the level of activity decreases, then we're biased toward dephosphorylation, and that's because of the different affinity of calcium calmodulin for either CAM kinase 2 or calcineurin. Okay, let's summarize our signaling here real quick. So let's look at some of the logic that we tend to use with these intracellular signaling pathways. So receptor tyrosine kinases. Okay. One of the things that you tend to see in signaling pathways is there's some sequestration of components. You take different proteins and you put them together. You put them near one another. Okay, and that's exactly what happens with receptor tyrosine kinases. The ligand binds, and that brings together these two subunits. That's what we see here. So the ligand is the red cherries. I don't know what the hell that is, but you see it's the red stuff on top. Those float on down and bind to their little receptor tyrosine kinase cartoons. You can see they're independent subunits. Once the ligand binds, they're put together. And now what you're doing is biasing the production of whatever it is this reaction carries out. And because when you increase the concentration of reactants, you're going to thus push toward the creation of products. Okay, and in this case, what we're creating are binding sites for other proteins. So we're bringing more proteins near one another so they can act on one another. So there's a lot of sequestration going on in these signaling pathways. By bringing the reactants together, we can produce products more efficiently, and by bringing different proteins together, we can allow them to interact with one another. Another thing that you see in these signaling pathways is amplification. Because we're using cascades of different proteins, we can amplify that signal because each kinase can phosphorylate multiple targets. And generally in signaling pathways, you have multiple kinases. And they're usually named very creatively. For example, MAP kinase 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 is the kinase that phosphorylates MAP kinase kinase. Makes sense, right? Because MAP kinase kinase is the kinase that phosphorylates MAP kinase. And MAP kinase is the kinase that's activated by mitogens. Anyway, because each kinase can activate multiple, can phosphorylate multiple targets, each one kinase creates many active kinases below it, and those active kinases then go activate many other kinases, so you get this amplification. You also get divergence of your signal, so each kinase is going to actually act on multiple targets, right? It might activate, 3 transcription factors or just the one. So you can get this amplification. You can also get many, many different targets affected by the divergence. And then finally, often forgotten is the feedback inhibition, right? That homeostatic controller that must be present in any signaling pathway. So if we start at the top here, we have the growth factor binding to the receptor tyrosine kinase. That brings the 2 subunits together. They phosphorylate one another. That brings a whole bunch of proteins up together. That then causes the activation of, RAS, which stimulates RAF. That's MAP kinase. MEK is MAP kinase, and ERK is MAP kinase. Anyway, once we get down to ERK, or MAP kinase, well, you'll notice, we have some divergence. There are a few different transcription factors that are activated. But notice the red inhibitory arrows coming back up. MAP kinase, or ERK, can phosphorylate its receptor and turn off that receptor tyrosine kinase. It can also turn off the upstream kinases. And so this allows the signaling pathway to turn itself off. So once we have activation of the receptor, if that signaling pathway goes as it should, if we actually have activation of the appropriate kinases, then they turn the whole system off. All right, and this creates brief bursts of signals, and that's what you want. You want to have a signal that's very strong, but also very brief. So only while the growth factor is bound are we going to have strong stimulation of this pathway. Once the growth factor is gone, MAP kinase is going to turn this whole thing off. All right, so we mentioned transcription factors. Let's talk about what they do. Right, this is kind of the ultimate change you can make in any cell, whether it be neuron or otherwise, is to change its gene expression. The way that we do that is by modifying the activity of transcription factors. All right, and transcription factors are just proteins that are involved with the process of transcription. Factor just means thing, so these are transcription things. Okay, in general, transcription is going to involve initiation, which is binding of RNA polymerase to the promoter, elongation, which is the synthesis of RNA from a DNA template, and then termination, the release of RNA from polymerase and the release of polymerase from the DNA. You see regulation occurring at the level of initiation. Once you start transcription, it doesn't make sense to stop it. The most efficient way of regulating transcription is at the level of initiation. Okay, if you don't want to express a gene, just don't even start. So initiation, that's where we're going to have RNA polymerase bind to the promoter. We have to position RNA polymerase at the appropriate location, and in order for that polymerase to bind, in order for it to find the location, it needs quite a bit of help. And so that's where the transcription factors come in. So there's this conserved sequence of TATA. That's the TATA box that you find in every promoter. And there's a protein that binds to this. TATA binding protein is going to bind at the promoter and then unwind the DNA to allow RNA polymerase II to bind. RNA polymerase II isn't going to bind there, though, unless it has the appropriate transcription factors to help it find its way. All right, and that's what we're seeing here. So at the top of this illustration, we have our TATA, T-A-T-A, shown in the DNA. That's showing you that this is a promoter of some sort. Tau binding protein binds there. It then sort of unravels the DNA a bit, and you can see that there are transcription factors that are getting recruited there to allow RNA polymerase, that big red jelly bean, to assemble at the promoter and then go ahead and actually carry out transcription. So the function of transcription factors would be to help RNA polymerase find the promoter. And they do this by binding to what we call cis regulatory elements. Cis meaning they're on the same strand of DNA, and regulatory elements meaning that they regulate the efficiency of transcription. There are enhancer sequences that will bind transcription factors called activators, and they will help facilitate RNA polymerase binding. Not every transcription factor is going to be helpful, though. Okay, and so some are going to be what we call repressors that bind to operator sequences and interfere with RNA polymerase binding. You should keep in mind that these cis regulatory elements can be very, very far away. Because DNA is flexible, these enhancers, even though they're very far away from the promoter, can actually become near them when the DNA simply folds over, and that's what we see in this illustration. That activator protein is bound to an enhancer pretty far from the promoter, but by folding over, now it's immediately proximal to the promoter. It can help recruit RNA polymerase so it can carry out gene expression. And this is just showing you the activity of repressors. So operators tend to be nearer the promoter, and when repressors bind, they're going to physically block RNA polymerase from binding to that promoter. And thus they're going to prevent transcription. Now, there are many ways to turn on transcription factors. So one way of doing this is to signal to the transcription factor to translocate from the cytoplasm into the nucleus. Okay, many transcription factors are hanging out in the cytoplasm, which is not where they actually function. The example of this would be NF-kappa B. This is a transcription factor that's normally held in the cytoplasm by this inhibitory protein. Once we have stimulation of intracellular signaling, in this case, if protein kinase C is activated, one of its targets would be that inhibitor, IKB there. Once that's phosphorylated, NF-kappa beta is free to move around the cell. It can move to the nucleus and thus associate with those cis regulatory elements. Sometimes proteins are only active when they form multimers, either dimers or, in this case, tetramers for this cartoon shown on the right. So the STATs are transcription factors that have to function as a multimer. So until they are phosphorylated, they remain in an inactive monomeric state. But when they're phosphorylated by receptor tyrosine kinases, then that phosphate group causes them to change their shape so that they can actually interact with one another, form that active multimer, and move then to the nucleus. There's also just direct activation by phosphorylation. You don't need to form a multimer or anything like that. The protein is just waiting for phosphorylation. The example of this would be CREB, or the cyclic AMP response element binding protein. CREB is going to bind to the enhancer, but it won't actually be able to interact or won't be able to help guide RNA polymerase unless it's phosphorylated. So only phosphorylated CREB can then bind with other proteins to help recruit RNA polymerase to the promoter. So in some cases, the phosphorylation simply allows the transcription factor to move, as with NF-kappa beta. Sometimes the phosphorylation is needed for the transcription factors to assemble, as is the case with the STATs, and sometimes the phosphorylation just turns the transcription factor on. That's all it is. Here's yet another example. Phosphorylation can induce dimerization. Oftentimes, transcription factors function as dimers. And what the dimerization does is actually create a larger binding site for the DNA. And so without this, dimerization, the transcription factors can associate stably with the DNA. Usually this dimerization is caused by the exposure of hydrophobic amino acids, which causes the creation of what we call a basic leucine zipper. That's where these exposed leucines cause the two monomers to fuse to one another, essentially through hydrophobic interactions. So phosphorylation can essentially cause the hidden basic leucine zipper to become exposed, and then the two monomers come together and form the active dimer. And that's what we can see here. Here's some inactive transcription factors floating around. You can tell they're inactive because they're gray, of course. They only become active once they are phosphorylated. That exposes that basic leucine zipper. They dimerize, and now they have a much larger binding site. They can stably interact with the DNA. Sometimes there isn't any need for phosphorylation. Transcription factors can simply be activated by external signals, like hormones. So steroid receptors usually have multiple domains. There's the transcriptional activating domain, there's a DNA binding domain, and then there's a hormone binding domain. So again, like other transcription factors, like NF-kappa beta, the steroid receptors are going to be held in the cytoplasm by some sort of inhibitory protein complex, and only when the steroid receptor binds to the hormone is that interaction disrupted, so that the receptor can move to the nucleus, generally dimerize, and then initiate transcription. Now, of course, here we've kind of painted a very simple picture where we activate a transcription factor, it goes and it activates one gene, and that's it. That's, of course, absolutely not at all what the cell is going to function like. There's usually going to be multiple transcription factors activated in sort of cascading fashion, so one after the other. First, you'll have the activation of what we call immediate early genes. These are transcription factors that are going to turn on other genes. So these are the first to get activated by those signaling pathways that we've gone over in this lecture. The immediate early genes then turn on the other genes by actually initiating the transcription of the transcription factors that activate those either delayed early genes or the then secondary response genes. So it's not just activating one transcription factor. First we activate the immediate early genes, and then they turn on a whole host of other genes by stimulating the expression of the appropriate transcription factors. CFOS is one that we should be familiar with. This is a very common immediate early gene that's measured usually as a marker for neuronal activity, because neuronal activity stimulates those immediate early genes. And so people will often look for CFOS as a marker of neuronal activity. And that's all they're showing you here. They're showing you the fold change in the expression of different transcription factors, showing you that CFOS is strongly activated immediately after stimulating neurons. Depending on which of the delayed early genes and secondary response genes are activated, this will then determine what's the long-lived change that occurs in the neuron. Are we generating more glutamate receptors? Are we stimulating the growth of the cytoskeleton, making the cell bigger? So the details depend on those secondary response genes. The immediate early genes are sort of very broad strokes, because they'll just activate other transcription factors. Okay, that about does it for intracellular signaling. There are many other kinases we could have gone over, but these are the main ones. If you have any questions on the material, don't hesitate to email me, and I look forward to seeing you in our next lecture.